Welcome back to the Neuroblastoma Parent Global Symposium 2022. My name is Antonia Palmer, and I am a childhood cancer research advocate from Canada and mom to a two-time neuroblastoma and thyroid cancer survivor. And I'm pleased to be moderating a session entitled CAR T-cell Therapies, Where Are We Now? So today we are honored to be joined by Professor John Anderson, Professor of Experimental Pediatric Oncology and Honorary Consultant Oncologist, Clinical Professor and Consultant Pediatric Oncologist at Great Ormond Street Hospital in London. Dr. Andres Hexy, Pediatric Hematologist Oncologist, Director of Cancer and Hematology Center at Texas Children's Hospital. Dr. Mark Yermarkovich, Senior Scientist, Division of Oncology at the Children's Hospital of Philadelphia. Dr. Rosa Wynn, the Pediatric Oncologist and Physician Scientist at the NCI Center for Cancer Research. And hopefully we will have Dr. John Maris joining us later at the end of the discussion, who is the Professor of Pediatric Oncology at the Perelman School of Medicine at the University of Pennsylvania and Division of Oncology, Children's Hospital, Philadelphia, sorry. So before I hand it over to our first speaker, I'd like to draw your attention to the chat function on the right of the screen, where you can submit your questions. There is the option to do so anonymously if you choose, and the questions will be addressed after all of the presentations have concluded. So now I'd like to hand the floor over to Professor Anderson to present on optimizing CAR T-cells against neuroblastoma. Thank you.
Thank you, and can you hear me, Antonia? Am I coming through?
Yes, we can hear you. Thank you.
Excellent, because I'm speaking through my phone and projecting through my computer because of a problem with the bandwidth. So thanks to everyone for getting it to work. But I can't find the little button for going forwards and backwards. Oh, I found it now. So these are my disclosures, and many thanks indeed for inviting me. So because I'm giving the first talk, I've made it quite a basic talk with an introduction to what T-cells are and what CAR T-cells are. And I think I would say the concept here really is that T-cells are cells of the immune system, which we all have in large numbers in our blood, which are professionals at fighting things which should not be in the blood. So they typically react against viruses, and they've evolved to be protect us from infection. But it's now known that they also protect from cancer. And indeed, part of the method by which cancer develops is by evading T cells and cells like this of the immune system. So they're very, very effective killers. And they can therefore become living drugs, drugs which not only are cancer specific, but have the capacity to divide, make copies of themselves, and expand to larger numbers. So CAR T therapy is a form of gene therapy, and the concept of gene therapy is genetic modification. So like genetically modified crops or genetically modified animals of other sorts, the concept here is that you put a gene into a cell which normally isn't present, and that gene changes its function. It actually works by making a new protein. Genes encode proteins, and essentially with CAR T-cell therapy, in simple terms, you're putting one gene in, which encodes for one new protein. That protein confers new properties. And of course, the properties we want to confer on the T-cells is a redirection of their killing. So the specificity of the killing is redirected towards cancer cells. And also these genetic modifications can sort of boost the function of T-cells in various ways. Okay. So how do we put a new gene into a cell? Well, the way which is typically done is by use of a virus. We all know about viruses. They infect people, and they infect people by infecting individual cells. There are lots of different types, but one of the types of viruses is called a retrovirus, and it functions by inserting certain genes into the chromosomes. So in this picture here, I don't know if my pointer is working. Well, I'll assume it is, and hopefully you can see that there is a yellow gene here, which is being replaced by an orange gene. and these are encoded within a virus particle. Here's a virus has essentially got DNA inside them. Viruses get into the cell, release their DNA, and that DNA gets inserted into a chromosome. And so this new gene goes into a chromosome, as a result of which this new protein is made that ends up on the cell surface and confers new properties to the cell. So that's the concept of what gene therapy is and how it works. So if we have one virus going into one cell, and the new gene, shown here in blue, gets inserted into it, into the chromosome, when that cell undergoes a cell division, you then have two cells, each of which has got the new gene in. And obviously, if these then divide, you'll get 4 cells. They've all got the gene. This is the concept by which you can expand the number of T cells. So now we think this is a T cell, it's a killer cell. It's not very good at killing cancer. It's infected by the virus outside of the body, and the new gene is put in. And then if you can trigger all these cells to expand to huge numbers, you get a very large number of cells, all of which got the same new gene in with the same new specificity for cancer. That's the concept of how it all should work. So this is the principle now of combining gene therapy and immunotherapy. We take a blood sample from the patient. You can collect the T cells from that blood sample, add the virus so they become genetically modified, then typically purify them so there's some sort of form of purification goes in, and then they get administered back to a patient either as an injection or as an infusion. Now, actually making these sorts of cell products is both complex and expensive, and it requires special facilities, which in themselves, building them is specialized. Many of these exist within pharmaceutical companies. They're essentially clean air facilities with special type of air handling. But certain academic hospitals also have their own facilities. This is called a GMP facility. Now, when we did our first trials, which is like going back 10 or 15 years when we started, it was much less sophisticated, but we did wear these sort of spacesuits and have to handle things in tissue culture laboratories in the context of very clean air. Increasingly, we're moving towards a slightly higher throughput approach. This is an example of the sort of machines available, and this is one of the approaches we use in our own hospital. It's essentially an automated cell processor. So you put the cells in at one end, add the virus, and the machine essentially performs all the complex techniques which we used to do by hand. And this potentially has the opportunity to bring the price down significantly. So I was privileged to be part of one of the early CAR T clinical trials, which we ran in London in our hospital, Great Ormond Street in Neuroblastoma. And it was it was certainly not the first time this had been done. And we have to pay credit to the great pioneers within the field, especially at Baylor College and Texas Children's Hospital. But we generated a novel antibody against GD2 because GD2 is a very good target for neuroblastoma. And this is what a CAR looks like. It's got an antibody, essentially, which is an anti-GD2 antibody at the end of it, some kind of projection which pushes it away. This is the cell surface, and now this is the inside of the cell. And if the GD2 is present on a target cell, this part, the brown part shown here, which looks like an American football, will bind the GD2, and it triggers the inside part to instruct the T cell to kill. And at the bottom, I'm showing essentially what happened to our first patients. Each line represents a patient, and this is the measurement of the CAR T cells against GD2, which we found in the blood. And so we went up to quite nice levels, and this high level seen here around like day 10 to 12 was associated with very high fevers, and we had to give medications to actually switch down the very strong immune response. And in a number of the patients, we did see a shrinkage of the tumours, but it wasn't complete, and it wasn't in all the tumour sites. But what was very disappointing from the trial, and wasn't necessarily what we expected, was that by day 30, 40, and so on, these T cells were disappearing. So the form of CAR T cell which we had made was not very well adapted in retrospect for long-term survival and long-term remissions in the patients. However, what was most important from the study and other studies performed in the field around the same time was we did not see the sort of awful neurotoxicity which we thought we might see in those patients. So a lot was learned. And with my colleagues, Robbie Maisner, who will be speaking later, and Paul Sondell, we recently thought about ways in which we could maybe try to improve CAR T-cells in neuroblastoma. And this is from a publication which recently came out, which is free online and light bedtime reading. And essentially this shows there's a selection of antigens as well as GD2, which could be good targets for neuroblastoma CAR T-cells. We're going to hear about GPC later on in this session. And ALK1 is one which my group have been working on, and a number of groups have been trying to target this antigen B symmetry because it's also expressed on a lot of other childhood cancers. But essentially the sorts of approaches which I think are available to try and improve the CAR T in the next sort of generations is by the engineering. So rather than a single gene for the gene therapy, putting in several genes to try and make T cells live longer, complicated things where we're putting 2 CARs in rather than 1. I think that is probably a little way from getting to the clinic in solid tumours. And then combinations, what other drugs and treatments can we give in order to get the T cells to survive longer? Of course, with clinical trials, one first has to find out that your T cells alone are safe and well tolerated. But then if they are, then potentially we could start adding agents to try and improve these things. And also changing the cell type. And so you'll also hear shortly about an alternative type of T cell called an NKT cell, which can be used with CARs. Now, although our data was a little disappointing, my colleague, Professor Franco Locatelli in Rome, has also been running and developing an anti-GD2 CAR T-cell trial, and it's not yet published, but actually will be published quite soon. And we're not allowed to show the very up-to-date data. In fact, I don't actually have access to it, but he did give me this slide from a year ago, which showed that the response rate in their patients was higher than we'd seen in our own trial. For some technical differences between the actual genes inserted in the trial we ran in London, plus compared to other GD2 CAR trials around the world. And maybe the differences in the design combined with the way in which the T cells are manufactured have contributed to what appears to be better response rates. And so this will be published soon, and it'll be very important and interesting to the field when we see it and can build on it. And this is one of the early slides, which again he shared me showing in their GD2 CAR T-cell, also well tolerated. They saw evidence of tumour shrinkage, and some of these tumour shrinkages were more prolonged than we had seen. And similarly, they had actually used this in some other GD2 positive tumours like Ewing sarcoma and osteosarcoma, and seen some encouraging clinical responses. So in London, we will plan to open a new GD2 CAR clinical trial. It's only a phase 1 study, which means that there'll be a small number of patients, and it's testing the safety of this. It's built on the same antibody which we used in the first trial, but it's added these 3 additional modules. So now it's a gene therapy in which rather than just 1 gene, there's 2 which go into the cell, and 2 separate viruses, in fact. And it's going to be run by a company called Autolus, and it's an example of the partnership between academia and industry, which is one way in which these potential therapies can become more affordable. The 3 modules are all designed to try and help the T cells, the CAR T cells, within the context of the inhibitory tumour environment to overcome all the inhibitory signals. So quite technical, but essentially they're blocking the brake systems which naturally exist in tumours to stop T cells from killing tumours. So I was only asked to give a very short introduction, and this is what my conclusion slide really is. I think in developing the field further, we have to be cautious because of a potential risk for severe toxicity. We can make T cells more powerful by putting more genes in and by adding to the therapeutic burden. But of course, as you increase the potency, there isn't a risk, as shown on the right-hand side, of increased toxicity. Also, as we kill more and more tumour cells, there's a risk that the antigen, that means the GD2 on the surface of a tumour cell, goes down, and the tumour cells with low levels of antigen or low levels of GD2 are the ones which escape and cause the relapse. So if we increase the responsiveness against tumour cells which have decreased or evolved with low levels of GD2, is the risk that the overstimulation we get in the CAR T cells can also lead to their eventual loss of function. That's something which we call exhaustion. So I think this tries to summarize the three sort of main areas in which we can need to fine-tune CAR T cells in the coming years in order to get them to live the correct length of time with the correct degree of specificity and avoidance of toxicity. So to evaluate all these things together requires a very sophisticated approach and a lot of international collaboration and working together. So these are my concluding comments. I think we've seen in the first trials of GD2 CAR T-cells in neuroblastoma that these cells really do have capability to shrink tumours. And importantly, there's emerging quite clear evidence that it does not cause the sort of awful neurotoxicity, that means pain, associated with tumour T-cell infusion, which at the start was a major concern and worry. We found that if we can get persistence of CAR T cells to be improved, then there is a real evidence emerging that remissions of tumours can be sustained. At the moment, it's very expensive to make these things and costs a huge amount of money. A recent trial application I submitted last week was costed at £4 million for 18 patients, just to give you an example of the sort of levels it is. And I think that we should be interested in whether we can target other targets as well as GD2 for other children's tumours, although GD2 remains a promising target for neuroblastoma. So that was all I was going to say in my short introduction, but I gather that we're going to be having questions later on.
Thank you so much, Professor Anderson. You're exactly right. We will have we'll have time for questions at the end of all of the presentations. But thank you so much for setting that stage so beautifully on CAR T-cell therapy. I'd now like to invite Dr. Hexy to the virtual stage to present on GD2 specific CAR and interleukin-15 expressing autologous NKT cells. Thank you.
Thank you very much, Antonia. I hope you can see my slide. Are you able to see the slides?
They're just coming up now. They're not in presentation mode just yet, though.
Okay, here you go.
Yeah, perfect. Thank you.
All right. Well, first and foremost, I wanted to thank for the opportunity to present. It's really an honor to be with this group. This is the first time, and I always found the discussions really stimulating and engaging. So my presentation is focused on the current phase 1 study and the emerging results testing the GD2 CAR NKT cells that co-express interleukin-15 and are evaluated in patients with neuroblastoma. These are my disclosures related to the platform and receiving funding from Athenex. So I think John had a fantastic introduction to CAR T cells and in general cell-based immunotherapies. And just to reorient us, about 10 years ago, there was a lot of excitement and still continues to be in leukemias, in hematologic malignancies, where several papers came out showing up to 90% complete remission rate. And now we know that even the longer-term survival, longer-term follow-up, even free survival, so being without the tumor, is about 50% in these very heavily pretreated patients who had otherwise uncurable leukemias. Well, unfortunately, that hasn't really been the case for solid tumors. There hasn't been that many studies either, but thus far, I think the field hasn't really reached the full potential of this therapeutic platform. And I think all of us on this panel feel that there are ways to reach similar efficacy. And how to get to that efficacy, there are different ideas. One of the ideas that we are really considering is just to stepping back, that are we sure that just bulk T cells are the best way to attack solid tumors? When T cells are a subset of white blood cells, but even then they have very different groups, some of them with more or less anti-tumor properties. So is there a better subset of white blood cells, a unique subset that would be perhaps really attractive to treat patients with neuroblastoma? And we believe that natural killer T cells might be such attractive immunotherapeutic platform, if you will, as opposed to T cells, which you can reach. This schematic represents how T cells interact with antigen-presenting cells. So T cells have this molecule T cell receptor on their cell surface that will recognize some peptides that are presented by these MHC molecules or antigen-presenting cells. So let's say a cell gets infected by the virus protein gets expressed on the cell surface, and the T cell comes and gets activated from it. NKT cells do have a T cell receptor, but all of us on this presentation share the same NKT cell receptor, which consists of the variable alpha 20 alpha chain, and it's preferentially paired with the beta 11 chain. And what's interesting is that it actually recognizes mostly glycolipid molecules presented in the CD1D molecule in an antigen-presenting cell. So why would that be relevant? Neuroblastoma is actually CD1D negative. However, there are some cells in neuroblastoma that support neuroblastoma growth that are CD1D positive. So why would NKTs be more attractive for neuroblastoma therapy? Well, one, NKTs can traffic to neuroblastoma in a chemokine. Certain molecules are produced in neuroblastoma, CCL2 and CCL20, and they can attract NKTs specifically to the neuroblastoma tissues. Once the NKTs are there, through this CD1D interaction, they target other white blood cells in the neuroblastoma microenvironment. So these other white blood cells are called tumor-associated macrophages, and those in turn support neuroblastoma growth. So by eliminating this supportive mechanism from neuroblastoma, NKTs indirectly target neuroblastoma by their innate properties. And if you look at outcome of patients, dividing them up by the level of NKT cell infiltration, it turns out that those patients who have more NKT cells do better. Their outcomes are better. So the question was, well, can we actually engineer CAR NKT cells so that they maintain their innate ability to inhibit neuroblastoma growth, and now, through genetic engineering, be able to directly target neuroblast cancer cells themselves? And this led to the current phase 1 study. It's called GD2 that is enrolling patients with relapse and refractory high-risk neuroblastoma. The preclinical evaluation led to moving a second-generation GD2 CAR into the clinic, and we also co-express another molecule called interleukin-15 to make the cells last longer and survive in patients longer, especially within the immune-suppressive tumor microenvironment. So how does this study run? There are two phases. The first phase is called procurement, and in that case, the cells are collected through an apheresis procedure, and the product is made. Once that's completed, the patients enroll on the treatment phase of the study, in which on the first 3 days they receive lymphodepletion with cyclophosphamide and fludarabine. The goal here is not to treat the tumor, but to create an environment where the infused CAR NKT cells would be able to engraft and expand and survive longer. And thus far, we completed 4 dose levels. And based on the results, now we are able to continue dose escalation for 2 additional dose levels. The primary objective of the study was safety, persistence, trafficking, and of course, even in the context of a phase 1 study, we were looking at anti-tumor responses. And this is kind of the schematic of how the study is done. So with that, I would like to conclude that we believe that CAR NKTs can be now manufactured to clinical scale in the GMP environment. And fortunately, they are safe, at least now, on 4 dose levels in heavily pretreated, relapsed refractory high-risk neuroblastoma patients. We do see in vivo expansion post-infusion. The NKTs can localize to metastatic sites, whether it's bone or bone marrow. We've shown that. There is tumor regression and 1 durable response already. And the expansion and persistence correlates with the CD62L frequency in the product. And perhaps there are ways to then enrich for these subsets so that all patients will start to respond nicely. With that, I want to thank everyone who contributed. Based on these encouraging results, now we have approval to continue the trial on dose level 5 and dose level 6. Most importantly, I want to thank the patients and families that commit to the trial and the referring physicians who believe that this is a good option for the patients. And Dr. Metalitza has been pioneering NKT cells and their study for the past almost 2 decades. I've been fortunate to work with him. There are several people who contribute, and I'll just leave it right there. Thank you.
Thank you so much, Dr. Hatsifa. Really beautiful presentation. Just like now to ask Dr. Yarmarkovich to join us to present on FOX2B peptide-specific CAR T-cells.
Hi, everyone. Can you hear me?
We can hear you and see your slides.
Great. So it's a privilege to be presenting to you. And excuse me if I'm a little congested, just getting over a case of COVID. So today I'd like to share with you our work on developing a slightly different approach using CAR T-cells. So as the previous speakers have nicely introduced, CAR T-cells have shown great promise, and I think this is really nicely illustrated by the story of Emily Whitehead that I think many of you are probably familiar with. So Emily came to CHOP with a previously incurable type of B-cell leukemia. She was given a single dose of CAR T-cells targeting a molecule called CD19, and this is Emily now just a few months ago back at CHOP. And what's quite remarkable is that when you look in Emily's blood, there's still functional CAR T-cells even 10 years later. And this is now being called a cure. So despite this amazing success, these successes have really been limited to largely liquid tumors, aside from some a growing body of case studies. And another type of immunotherapy called immune checkpoint inhibitors have been highly effective in another subset of tumors, namely those with a high burden of mutations. And really this excludes the majority of pediatric cancers, which are on the lower end of the mutational burden spectrum. And when we roughly group these tumors as low mutational solid tumors, you can see that for the vast majority of cancers, there are still very few effective immunotherapies. So why is that the case? And there are several reasons that go into this, and I'll just talk about one of them, and that's the limitation of tumor-specific targets. So what we think makes a good tumor-specific target is a target that's exclusively expressed in cancer cells, but also required for the biology of the cancer to continue its growth. And you can see here the landscape of some of these targets in and across different cancer types. And the majority of the CAR targets have been those that are on the surface of the cell. And what we know is that the proteins that are on the surface of the cell represent only a small proportion of the total cell protein. And of these, very few are actually essential to the biology of the cancer. Whereas what we know is that the majority of the most well-characterized drivers of cancers actually reside within the cell. And to date, these have been very difficult to target using CAR T-cells. And so just to give a little introduction to a component of the immune system that actually presents the intracellular contents of a cell on the surface of the cell, very briefly, this is a system that likely evolved to present to the immune system viruses that had infected cells and that were replicating within the cell by degrading these viral proteins into small fragments called peptides. Some of these peptides are able to be picked up by a molecule called MHC, also known as HLA. And when these small fragments are picked up, they're transported to the surface of the cell. And that's where the native T cells survey this complex of the peptide with the MHC. And if their receptor recognizes that peptide MHC complex as foreign, that will induce cell killing. Now, the pathway that presents these protein fragments doesn't discriminate whether a protein is derived from a virus or if it's derived from the proteins of the cancer cell. So many of the self proteins are actually presented on the surface through the same process, but those that are non-mutated and not recognized by T cells are ignored by T cells. So some types of these mutations that lead to peptides that presented on the surface that can be recognized as foreign can in fact induce killing of a cancer cell. But that really requires a high burden of mutation within the cell. And as I mentioned before, most pediatric cancers really don't have that type of mutational burden to trigger these. Nonetheless, we thought that perhaps drivers of these cancers could actually generate peptides that could be presented on the surface of cancer cells. And we wanted to test this hypothesis of whether these drivers of neuroblastoma could be presented on the surface of, from within the cell, on the surface of the cell as non-mutated peptides. And if so, could we target these using CAR T cells? And so this is kind of a diagram describing our work that was recently published, going from target discovery to target validation, our CAR receptor engineering process and screening platform. And ultimately, this has now led to preclinical studies that are leading to a trial we're hoping to open sometime next year. And I'll try to run through several of these different steps. So first, we started by combining data sets of sequencing, DNA sequencing and RNA sequencing, both from a set of neuroblastoma tumors as well as a set of normal tissues. And we combined an experimental approach with a computational approach to what we identified were about 9,000 of these peptides that are presented on the surface of neuroblastoma cells. And through this computational filtering, we were able to narrow that down to about 56 that we thought were highly specific in neuroblastomas. And to give you an example of one of these, this is one of the well-known drivers of neuroblastoma, a gene called FOX2B. And shown here is the expression of FOX2B in the first column in neuroblastoma. And then in the subsequent columns, this is the expression of the same gene across about 1,600 different healthy tissues that are grouped by different organ sites. So what we see is that FOX2B is very highly expressed in the tumors and then completely absent in the healthy tissues. And this is exactly the type of profile that we're looking for this type of target. And just to contrast that to a target you've heard about, GD2. GD2 is also overexpressed in neuroblastoma, but in this analysis, we also see that it's expressed in brain as well as nerve, and that's consistent with some of the toxicities that are seen in the clinic. And this isn't to knock GD2 as a target. We think that ultimately this will be a complementary approach and combining these different strategies. But we think that a target like this will really allow for an expanded therapeutic window in CAR T-cell dosing. So now that we've found this target, I just wanted to highlight the challenges of developing a CAR T-cell receptor for a peptide that's presented on the MHC. So this is an actual image taken from X-ray crystallography of the FOX2B peptide. So this is a small fragment of the FOX2B gene that's 9 amino acids long, and this is the FOX2B peptide in the context of the MHC molecule that presents it. And what you can appreciate here is that the portion of this complex that's derived from FOX2B represents a very small surface area of the peptide MHC complex. And that's only about 2% of the surface area of this molecule. And what's very important is that when we develop a CAR receptor, that receptor has to only bind to this FOX2B peptide, which is the tumor-specific fragment, and with no cross-reactivity against the much larger MHC molecule. And this is a very technically difficult challenge. And I liken it to identifying a needle in a haystack. And without getting into the technical details, we start with a library of antibodies that are ultimately converted into CAR receptors. And these libraries are very large and contain a very diverse set of antibodies on the order of about 100 billion. And what we're looking for is the one single receptor out of these that has the exact binding properties of being able to recognize our FOX2B tumor-specific peptide and not cross-react against the MHC complex that's also co-expressed in healthy tissues. And in short, what we've developed is a process that combines experimental approaches with computational techniques to really narrow down this pool of 100 billion receptors to what we think are the top 10 receptors. And then we can empirically test those for the ones that have the exact binding properties that we're looking for, and ultimately identify that single needle in a haystack. And I kind of use the analogy of lock and key in the receptor that's able to recognize that peptide and bind it very tightly, but in a very specific manner. Now, to kind of illustrate what our lead receptor that came out of the screen looks like, I just wanted to show a couple of videos of this CAR receptor killing neuroblastoma cells. So on the left, we have two target lines, which are the neuroblastoma cells. On the right are two non-target cell lines. So I'll show you a video of the killing, and you can see that the killing is marked in red. So really what we're looking for is a CAR T-cell construct that kills all of our neuroblastoma cells and at the same time has no cross-reactivity to these non-target cells that have the same MHC type. And that's exactly what we see here with this lead CAR T-cell, is that we get complete killing of our neuroblastoma cells and no cross-reactivity. So encouraged by these results, we then tested these in patient-derived tumors that are grown in mice. And this is an example of two of these lines. And you can see plotted here are the growths of these tumors over time. And these two lines show the growth of untreated mice, as well as those treated with T cells that don't have the CAR receptor. And now shown in the blue and red are the same, the mice that grow the same tumors, now treated with these FOX2B specific CAR T cells. And what we were very excited to see is that these CAR T cells are able to completely eradicate these tumors in mice. And so we're very cautiously optimistic about now moving these CAR T-cells to the clinic. And just to give you a snapshot of where we're currently at, we're now performing safety studies to file an IND with the FDA. We've now completed manufacturing of our clinical grade DNA that will go into a clinical grade vector manufacturing process that started just a few weeks ago. And we're hoping to file as a new drug with the FDA. And importantly, one of the nuances of this type of approach is that the MHC is a highly divergent gene across the human population. In fact, it's the most polymorphic gene in the human population. And because of that, each patient will have a different HLA type that presents different sets of peptides. So far, we found that the FOX2B approach we think will be applicable to about 30% of neuroblastoma patients. And we're actually working on ways of expanding this to a larger proportion of patients, but also developing specific CAR T-cells using the same approach for those other 70% of patients. And we think it'll really require 3 to 4 of these types of CAR T-cells to be able to cover the entire patient population. So to summarize, we've developed, we're trying to address the problem of limited application in solid tumors of CAR T-cells that's largely limited by tumor-specific targets. What we've done is develop new technologies that identify new sets of highly specific tumor targets, those that are derived from inside the cell presented on the surface through MHC. And these targets are very difficult to develop CAR T-cells for. We developed additional technologies that allow us to identify these ultra-rare CAR T-cell receptors with the correct specificity profiles. And this allows us to identify a single receptor out of 100 billion with the correct properties. We've shown that the CAR T-cells targeting FOX2B show complete killing of neuroblastoma tumors in mice, and are moving this into the clinic in the next year. So as I said, we're expanding the strategies to expand the addressable patient population, both with the FOX2B CAR, but also developing additional CARs that will cover the other patient population. And we're also moving this approach beyond neuroblastoma now into other pediatric cancers. And so this has really been a large effort, both within John Maris's lab and across Penn and CHOP, but also really an international effort with collaborators in Australia and Germany and across the country. And this is just showing our funding sources. So I just wanted to put my email up in case anybody has questions. And then this is John Maris's email as well, whose lab I've been doing this work in. So I will stop it there. Thank you very much for your attention.
Thank you so much, Dr. Yarmarkovich. Certainly appreciate the challenges of presenting when you're not feeling well. So thank you so much for being here and for that really excellent presentation. I'd like to now ask Dr. Nguyen to join us to present on the GPC2 CAR for neuroblastoma. Thank you.
Yes, thank you very much for inviting me to be part of this symposium. It's always a great opportunity when we as basic researchers and clinicians can come together with patients, families, and the greater neuroblastoma community to share our vision and our data in the lab. So I was asked to talk about GPC2 CAR T-cell therapy for neuroblastoma, and particularly the work that we've done over the past 5 years in the pediatric oncology branch and the National Cancer Institute. And I wanted to start by putting up the schema on the left-hand side to show you all the different targets that have been and are currently of interest for preclinical and clinical CAR T development. And it's quite an interesting list because some of these targets are really mostly in neuroblastoma and maybe a couple more tumors of childhood, but there are also other candidates like B7H3 and GPC2 that are expressed in a variety of childhood tumors and therefore of particular interest to us. GPC2 belongs to a family of proteoglycans. There are several members in there, and the second member is GPC2. And it was really Dr. Mitchell Ho, who is a tenured investigator at NCI, who was one of the first ones to describe the expression of GPC2 in neuroblastoma. And his lab not only did that, but also really showed that GPC2 gives cues to the Wnt signaling pathway, which is activated in neuroblastoma cells to drive neuroblastoma progression and growth. And then really over the following 4 or 5 years, his lab together with Carol Thiel Labs, Javed Khan's, and my recent lab have put a lot of effort into developing a CAR T that can target GPC2 and optimize that CAR for clinical translation. And we were certainly not the only ones. You also had dear colleagues at Stanford and CHOP who are still very interested in this target and have published their findings in a variety of different papers over the years. However, I think it's important to mention that each of us use a different binder in CAR, and I'm going to use the next 2 slides to explain what that really means. So if you remember what Professor Anderson had told you about the structure of a CAR, it's basically an antibody that is stuck to the surface of a T cell and then helps the T cell recognize the cancer through a specific antigen and further signals downstream to activate that T cell and redirects it to kill the tumor cell. In our instance, our CAR is derived from an antibody called CT3. And it's a very interesting antibody because although it targets GPC2, which comes in different forms, which are shown here in this graph, it actually specifically targets exons that are found in tumors. So as I mentioned to you in this graph, you see 5 different isoforms. So these are slightly different forms of GPC2 that can be found in nature and in disease. And the 201 isoform is mostly present in tumors, whereas normal tissues express the 203 isoform. But as you can see very clearly, indicated by these red boxes, there are certain exons like number 3, 7, 8, 9, and 10 that you do not see in the 203 isoform, but mostly in the tumor isoform. And our antibody CT3 and our CAR target exon 3 and 10, so they are really even more tumor specific. And that is of advantage because if you think about toxicity or cross-reactivity, you do not want these CARs or antibodies to cross-react with healthy tissue if possible. I am a T-cell biologist and cytokine biologist by training, so I think it's important for me to at least mention also the importance of antigens in this context. What I mean with antigen density is that a cancer cell can have various levels of its target expressed on the surface. So if there are a lot of antigens on the surface, that's actually good for the CAR because as it hovers over the cell and tries to find the target to engage with it, it's easier if you have a lot of antigen or target on the surface. And that will help to activate the T cell and trigger killing. However, almost like a disadvantage or minor downfall of GPC2 is that it is expressed at low densities and low levels compared to GD2, for example, or B7H3. And those levels are in GPC2 up to 10-fold lower than what we see in those other targets. And so it really requires CAR engineering for optimal CAR function. And I put this cartoon up here for you to just show you on the left-hand side, the general CAR structure. So in red is that binder sequence, which is derived from the antibody. So we did not touch that, but we did modulate the hinge, the transmembrane, and the endodomain of these CARs to see by modulating that if we could optimize the kill function of the CAR. And then on the right-hand side of this graph, you see the different iterations of CARs when you modulate or change these different domains. And they're really grouped in generation or classes. And this should serve like basically as a cartoon, so you can see how many different varieties and flavors of CARs there are. As we embarked on this process to optimize our CAR, we really went through all of these different versions, but in the end decided to narrow it down to 3 distinct CARs to do head-to-head comparisons. And on the top of this slide, you see the structures of these 3 different CARs that we compared. The orange CAR is the one that we first published in the Cell Report Medicine paper and serves as a reference, but we have tried to improve it by generating this blue and the red CAR, and they, as I mentioned, vary in their hinge, transmembrane, and co-stimulatory domain. So for us to do head-to-head comparisons, the first thing we did was to produce T cells that had either one of these CARs on their surface, and we would co-culture them with tumor and see how much cytokine the T cells would be able to produce, so to speak, as a measure of function. And we co-cultured not only with tumor cells or neuroblastoma cells that had GPC2 on their surface, but what we also did, we stripped those cells of GPC2 and called them GPC2 knockout cells. So what you can see in both of these graphs on the bottom, where we measured interferon gamma and granzyme B, which are 2 cytokines and molecules that T cells produce, is that T cells alone or in co-culture with GPC knockout neuroblastoma cells really only produce a little amount of cytokine, but these black bars really shoot up when they see their antigen. So it's an antigen-specific effect. However, a little bit disappointing at that time was that there was really not a whole lot of difference between these 3 different CARs that we compared. But to do our due diligence, we actually proceeded and continued with our head-to-head comparisons also in animals. And here I want to draw your attention to the left upper graph, bar graph, where we used a very treatment-resistant patient-derived xenograft. And we implanted these cells into the adrenal glands of the mice, let them engraft with the tumor for 3 weeks, and then treated them either with control cells, which are called untransduced mock, or these 3 different CARs. And what you can see is that the controls really did not do much. These animals after 4 weeks had like a tumor burden, which was about 2 grams, which equals 20% of the body weight, which is quite a lot for a tiny mouse. But when you do give them CAR T cells, you can see that there is a moderate effect with the orange and blue CAR, though there was not a real difference between those, and a much stronger effect with the red CAR. Moving forward then, encouraged by this, we decided just to compare the unpublished and new CARs, which are the red and the blue CAR. And we did that in another tumor model that we used, which was an IMA5 model, which is a cell line, a neuroblastoma cell line, which was also implanted into the adrenal glands. And we let these mice engraft for 3 weeks again, and then started treatment. And on the bottom of the slide, you can see very nicely the responses. So these are graphs that are derived from scans which are similar to PET scans, where you can basically track the tumor growth over time. And you see that the mocks and basically the T cells without a CAR did not do much, and the animals came off study due to tumor progression by 5 weeks. But we had a very profound effect with the red CAR, where you see that the tumor signal was entirely ablated in all of these animals. And the blue CAR also had some animals that had no detectable signal anymore, but also some escapers where animals ultimately succumbed to their disease. And we really repeated this several times and also did survival studies, and were able to recapitulate these findings, demonstrating that the red CAR was really the winner across those 3. On this last data slide, I'm just going to dive a little bit into why that is, because we wondered what could be potential causes for such better effect. And the first experiments we did in my lab was to label the T cells so we could track them in the mouse after injection. So these signals that you see on the left-hand side that are highlighted in the mouse are basically the T cells that light up. And you see very nicely that after 24 hours, most of these cells end up in the lung. That is the anatomical route when you inject them into the vein, tail vein. So they first end up in the lung. But over the next couple of days, they really move out of there into the bone marrow and then ultimately into the tumor, which I implanted into the right adrenal gland and then the spleen on the left-hand side. And all of these CARs expanded beautifully, while the MOX really did not expand and did not home either. On day 7 then, we took these mice and looked at the tumors and evaluated the T cells that were homed and that were present in the tumor. And we found that most of these T cells and CAR T cells particularly really expanded and had a large fraction of CD8 effector cells shown in red in these upper plots on the right-hand side. And we did that with 2 different donors, which is very good because you want those type of T cells in the tumor because those are the tumor killers. But again, there was really not a whole lot of quantitative difference across these CARs. But when we did look at the transcription profile, so the gene signature of the red CAR and compared it to the orange and blue CAR, we really found that there were quite significant and important genes that were upregulated that play important roles in the proliferation and homeostasis of T cells, in their migration ability, as well as their function and cytotoxicity. So although there was not a big quantitative difference, there was definitely a qualitative one, which could explain why this CAR is doing so much better. So these were just snapshots of data that I showed you, but altogether the experiments that we've done rigorously in the lab really helped us identify a very potent anti-GPC2 CAR for the treatment of patients with neuroblastoma. So while I mentioned to you that there are 3 different centers in the US that are currently focused on GPC2 CARs, it's really important that all of these CARs go into clinical testing to see which ones is better and performs best, since all of them have different binders and are derived from different antibodies. We at the NCI plan to conduct a clinical phase 1 and 2 study, tentatively opening in Q1 of 2024 and offering this very promising therapy to patients. And I want to conclude by just pointing out that this is really a team effort. So there are many people involved, people in my lab, Riona Okada and Jeshka Reyes-Gonzalez, my 2 postdocs who have done a tremendous amount of work, but also Carol Thiel with her 40 years experience in tumor biology that has really complemented this work. And then my collaborator also and colleague, Dr. Ming Sung, with whom I've done most of this work. The whole lab with Dr. Mitchell Ho and Nan Lee, the whole POV leadership, and the Rupin lab who's done the biocomputational work, as well as our other core facilities with SCAF and CCE, who have helped us with the clinical translation process. And last but not least, of course, our patients and families who inspire us to do better. So thank you so much for your attention.
Thank you so much, Dr. Ray. That was a really excellent presentation. So I'd like to invite all of the speakers back to the virtual stage, and we will answer some questions that have come in from the audience. I'm certainly looking forward to having a great discussion about CAR T-cell therapy and neuroblastoma. All right. And Dr. Anderson, I'm going to ask this first question of you. So when a treatment is in a very early trial stage, many of those patients are ones where they've received a lot of treatment and where other treatments have not worked. For these early trials, how do you properly interpret the data in light of all of the therapies that patients have been through?
Okay, thank you. Can you hear me? Because I've just had to reconnect?
Yes, we can hear you.
Good. My Wi-Fi is still not working well. I think this question is saying, addressing the issue of affecting phase 1 studies, which typically performed on patients with multiply relapsed or disease, more typically than in primary refractory disease. They've had a lot of prior treatments and resistance to other therapies. I suppose the main difference here, though, is with the CAR T approaches, we're targeting both with the FOX2B type MHC approach and with the solid, the surface antigen approach, we're targeting molecules which are likely not to have been affected by prior chemotherapy, radiotherapy, and so on. There are some caveats to that and some interesting new findings about how some of these antigens are changed as a result and molded as a result of therapy. But I would say by and large, that is not the case. And it's interesting, when we were first looking at the GD2, a major concern we had was that we might have a resistance through either downregulation of GD2 by patients having previously received GD2 antibodies, or indeed there might be immune responses against anti-GD2 antibodies, which is why we selected to use a different antibody targeting GD2 within the CAR. But in fact, there's relatively little downregulation of GD2 as a result of anti-GD2 antibody therapy, but it is a real phenomenon. I think that's what the question was getting at.
Thank you so much. Dr. Yarmarkovich, we have a couple of questions around FOX2B for you. So one of the questions that is first asked is, do all neuroblastoma tumors express FOX2B?
Yeah, so as far as we can tell, all of the neuroblastomas we've looked at do in fact express FOX2B, and it's actually used as the diagnostic marker for neuroblastoma. And when you look in the data set of knockout libraries using CRISPR to look at which genes the cancer really relies on, FOX2B comes up as number 3 in that analysis. So it's the third highest ranking dependency gene in neuroblastoma.
Okay, great. And in terms of the trial that you're planning for next year, what do you foresee as being the conditions for that trial, how it will proceed, and the question also asks, where will the trial be held?
Yeah, so the criteria will be, as I mentioned, the FOX2B is really limited by the HLA or MHC that presents it, and so we've shown that the CAR can target FOX2B presented across multiple HLA types, and we're still testing the extent of that. And we're hopeful that we can get somewhere between 30 and 50% of the population with the FOX2B CAR. So that'll be really an important key criteria, is the HLA type of the patient. The initial trial will be opened here at CHOP, and if we see a good result, I think there's a plan to expand it through the PIDDN network, which is the Cancer Moonshot team across other sites and across the country.
Thank you. And I think I'll put this question to the floor, and whoever would like to answer it first, but I think we might have multiple answers for this one and responses. But how will it be determined which CAR approach should be prioritized? And can data from one CAR T-cell clinical trial be compared against others? Maybe Dr. Hexie, I'll get you to answer first.
Sure. Yes. It's an interesting question, because I think one of the challenges in the field is that, fortunately, the number of relapse refractory neuroblastoma patients is shrinking. The upfront therapies are working for the majority. Needless to say, there is a number of patients who need therapies. And as you can see, just by this panel, you listen to, at least 4 different approaches. And once we start to divide up these shrinking population of patients, it becomes really challenging to know which is the best one. And I don't have a good solution to it. I mean, right now the standard is that you go with these new trials into a phase 1 stage when you try to establish safety. And once that's done, usually it's a phase 2 assessment. In cell therapies, that's often just a direct extension of further enrollment on the highest dose level or maximum tolerated dose level. And then after that, one can apply for a drug approval. And then after that, you can run comparison randomized control trials. I'm not sure, given the limited number of patients for high-risk neuroblastoma, that by the time all these studies report out the data, that's going to be still a feasible path forward. It is a challenge. I think 10 years ago I would have completely agreed that the more is better, the more choice, the better. The problem is that then these trials, enroll for a very long period of time. We don't really get the data. So to go into in a multi-institutional setting, even for phase 1s, I think is an important part. But beyond that, I think the field needs to kind of combine forces to really, get the best immunotherapeutic approach to the patients at the fastest possible way. I hope that was a reasonable answer.
Yeah, it is definitely reasonable. Would anybody else like to answer that one as well?
Maybe I can just add to what Dr. Hexie just said. You're basically dealing with a living drug with a lot of different variables, not only the patient variables, since we're using the patient's own T cells, but also the manufacturing platform, the type of virus, the type of DNA that we integrate into the T cells to make them express the CAR. So these are all variables that make it from a comparison standpoint maybe difficult to do these head-to-head comparisons, even looking at different centers and how patients are treated there. But I think still from each of these trials with very carefully designed correlative studies, we can all learn from them and incorporate these findings into upcoming trials. So that I would think is the best way to learn and improve that.
Thank you. Dr. Wynn, the next question is for you. Are there other centers that plan to initiate trials that are targeting GPC2 in the next little while?
Yeah, although I'm not probably the best person to answer this, I do know that CHOP and maybe Mark wants to comment on that, CHOP and Stanford both plan to open the trials. In terms of their timeline, I am not sure. I can just speak of what we at the NCI do. So we're in the middle of performing the IND enabling study since this is a first in human CAR. We have the funding. It's very generously funded by the Moonshot Initiative. So we have the money to produce the drug and also the infrastructure here. But it's important that we jump through all the hoops of safety and regulatory, requirements to make it safe for patients. So that's why I put up a quite generous time estimation of Q1 in 2024, but it could be earlier than that.
Dr. Yarmarkovich, is there any plans for CHOP at this stage?
Yes, there is. So the GPC2 trial is it's actually slightly ahead of the FOX2B trial. And my understanding is the plan is to start enrolling patients mid-2023.
Fantastic. Thank you. Dr. Anderson, Professor Anderson, you have the next couple of questions. You talked about the risks of toxicity. How are these currently handled in the clinical trials? The question also goes on to say we don't want to see our children suffer. Supporting them through these therapies always needs to be a major priority.
That's a great question, and thank you to the questioner. I think there's two main points. These are always evaluated in the context of phase 1 clinical studies, at which the design, a lot of thought goes into the design so that the first child to receive the first drug will receive a lower dose. The lower dose may be less effective, but that's done as predominantly as a safety concern. And then the design of how the escalation through doses is done is now designed in what we call an adaptive way, so that as quickly as is safely possible, children will, the cohorts of children receiving different doses will be accelerated. So the emphasis there is on safety, but also on not necessarily having to give large numbers of children a non-effective treatment. When you're dealing with a first-in-child, first-in-human type study with a new agent, of course, we all know anything could happen. And so the second criterion, of course, is the eligibility criteria. And here, typically, we always require a child to have exhausted known curative potential approaches for treatment, and also to have a performance status, which is, and life expectancy, which is reasonable. In other words, we don't want to be giving potentially very toxic agents to children whose life expectancy is below, typically around about 2 to 3 months. So it's really that's an unsatisfactory response to a very important question, but essentially it's related to the design of the trials and the support committees and data monitoring committees which go into to scrutinize and ensure quality.
Right, and I guess part of what you're also designing into the trials are the sort of off switches. Is that right as well? Yeah.
I mean, not all CARs going into trials have off switches, and some recent approvals have not had them, but many do. One of the reasons for not including them is the off switches for a lot of the toxicities been quite well worked out through the experience in the leukemia field. So the sort of drugs which are needed to mitigate some of the immunological toxicities, like very high fevers and things, are well protocolized now. But there are, as you say, a variety of safety switches which involve drug-induced off switch of the CAR.
I think a concern among families is certainly around the neurotoxicity of a CAR T-cell therapy. I know you've talked about certain CARs not having the neurotoxicity, off-target side effects. Why is it that we are seeing some CARs having neurotoxicity side effects and others where we don't see that? Is there an understanding of that?
Right. I mean, can I address this one?
Yeah, absolutely.
I think it's really interesting, but I think we have to be cautious about generalizing the findings from leukemia to across every other cancer types. One is the toxicity part. I think the neurotoxicity component is still highly debated in the field, but there are publications in high impact journals suggesting that CD19, the target antigen, might be actually expressed in the perivascular, so right, beyond the little vessels in the brain, and maybe that's causing this inflammatory response, which that would not be the case in the context of GD2 or GPC2 and so on. I think in general, it's been a theoretical risk, but I have not seen any similar neurotoxicity in solid tumor patients thus far, that has been reported in leukemias. I'm sure maybe there is some, but it's the toxicity profile, even in patients who get CRS with solid tumors, is very different, at least in my experience and what I've read, than in leukemias, lymphomas. So I think we have to be cautious about generalizing that it's going to be the same. And in one of our other trial, when we treat liver tumor patients with a different receptor, the whole CRS profile is mediated by different cytokines too. So I think we just have to look at them differently. Look at each disease with each CAR slightly differently. Sorry, John, I think you were trying to say something too.
No, I was holding back, and I have nothing to add to a brilliant answer.
So one thing I do find curious about the GD2 CARs is with GD2 immunotherapies like dinutuximab, unitoxin, you see pain as the predominant side effect. With the GD2 CARs, we don't necessarily see that as the predominant side effect. Is there an explanation for that?
Yeah.
Andres, you go ahead.
Sure.
Yeah.
So I think, again, it's just trying to generalize findings from a totally different modality is where we have to be careful. So I think the pain that is shown with antibody-based targeting is mediated by different mechanisms. I think complement-dependent mechanisms have been known to be the primary cause, and that component of the immune system is not engaged when you are using CAR T-cells. So I think GD2 mediated neurotoxicity remains a risk, but thus far, I think it's a theoretical risk only. I mean, there have been now several trials targeting GD2 with chimeric antigen receptor T-cells or NKT cells in our case. And I wouldn't say it's not the predominant. We haven't even detected any. So I'm starting to feel pretty confident that maybe this is not something that we will encounter in the context of cell therapy. Of course, studies are still dose escalating, and there might, as we are increasing the potency of these, maybe there will be severe toxicities. But thus far, that has not been, at least our experience at all. John, what do you think?
No, I agree. It's exactly what I was going to say. The only thing I'd add is, the recent experience of using GD2 CAR T-cells in brain tumors, some of the work of which has been published, again indicates that there's a lot of toxicity, but it doesn't seem to be neural toxicity, pain toxicity associated with direct targeting of GD2 in neural tissue. So it is encouraging, I think.
So, Professor Anderson, you talked about how expensive CAR T trials are to conduct. Any thoughts on how this, and whether it's the point of care, point of production systems or others, but how is it possible to be able to deal with the challenges of these high expense health technologies, but being able to make sure that there's availability of these clinical trials and across Europe and North America and really around the world?
Sure. Thank you. Yeah, I mean, I'll give a European answer to the question. So the first point is that to roll a trial out to large numbers of patients, first of all, need success in phase 1, indicating some evidence of activity and benefit to patients, such that you actually want to evaluate it in larger numbers and give larger numbers of children the capacity to participate in clinical trials. So that is obviously the goal, and it may be that we're not as far away from that as we thought. So that brings us on to the second part. Assuming we've got a product which there is sort of an international consensus that it is working and worthy evaluation in large number of patients, how do you make it affordable? Well, the first part of the answer to that is international collaboration. Within Europe, this will come through organizations such as SciPen and ITCC, who really serve to coordinate efforts. You won't be surprised to know that we have had many discussions exactly about this, of how it might be possible to work together in terms of coordinating efforts, learning maximally, and getting the sort of funding you need. So those sorts of discussions are very much underway, shall we say, although it takes a long time to produce these sorts of trials. The second part of how we make it more affordable and realistic is, I think, automation and working within academia. So the cost of making CAR T-cells from the pharmaceutical industry is extremely high, but certainly within our own institution, we've been able to do things at a slightly cheaper way. And there's an economy of scale whereby if you're working together, sharing virus production, for example, which is one of the most expensive parts for a larger trial, that can bring the costs down. The complexity is that you would need several manufacturing sites, probably in different locations, working to exactly identical protocols and standard operating procedures, and there's a large level of bureaucracy around the standardization of that. But that's I'm sure where within Europe certainly we'll be aiming to be heading.
Dr. Nguyen, I saw you shaking your head in agreement. Is there anything you'd like to add to that one?
No, I think what Professor Anderson said was exactly on spot. Also in North America, I think we're mostly struggling with getting GMP grade vector and getting in line for producing virus. And I hope that as more trials roll out and maybe the guidelines become less stringent, because we will have more experience with the production and with the products, that this will be more streamlined and easier and better accessible to smaller institutions. I think most of the larger institutions have their own facilities to produce, like NCI, Stanford, CHOP. But it's and obviously Dr. Heckshey's institution as well. But I think it's a challenge for smaller institutions that do not have these resources.
Absolutely. Dr. Yarmarkovich, I'll ask the next question of you. So if CAR T-cell is successful in reducing disease, but does not produce a complete response, what type of treatment would be needed after? Are there limitations of what can be used after CAR T in order to preserve CAR T cells?
I think maybe that question's better taken by one of the clinicians, but I think within the treatment of a single patient, I wouldn't be able to answer that, but generally speaking, I think if we see that a particular CAR T-cell construct fails, I think there's approaches that can be taken to either boost the activity of the CAR T-cell within the tumor, or taking kind of a combined approach of dual targeting if the mechanism of resistance is through downregulation of the target, maybe finding a combination of targets that the cancer cell can't tolerate. That's another alternative strategy. But within the course of a single patient, maybe somebody else could comment on that.
Dr. Hexy or Dr. Anderson?
Sure. I think it really depends on the individual patient. If many of the patients who come on CAR T-cell therapies, they will have had a variety of upfront treatments, and then most of them will have several salvage regimens. So if there is something available known to have anti-tumor activity, of course that will be the first one to consider. Or other investigational trials, I think those would be the ones. What would be what drug or drug combination would not eliminate CAR T cells? I think that's a harder one to figure out. Conventional chemotherapy will have negative effects on CAR T cell persistence, and they will wipe them out. Theoretically, one would speculate that maybe checkpoint inhibitors would be something to consider. But in general, checkpoint inhibitors have been quite disappointing in solid tumors, including neuroblastoma. So I don't know if that would be something that people would want to consider. I think it's an individual choice depending on what the patient had and what other investigational opportunities are available.
And I guess another thing I could add to that is, we're looking at approaches kind of analogous to a vaccine, where we, if the mode of failure is through exhaustion of the T cells, where we can give kind of a small level of the tumor target to reinvigorate the existing CAR T cells. That's something else we're looking at.
Absolutely, thank you. This is a question for the whole panel, but also really directed for Dr. Nguyen. So are there any comments about accepting international patients on CAR T trials at your centers? Some sites don't permit international patients. The NCI does not charge for treatments trials. And just curious if your trials at your centers will be offering the allowance of having international patients on the trials.
Yeah, here at the NCI, we definitely will offer that to the patients coming onto the GPC2 trial. I think each patient that will be referred will go through the same evaluation criteria and screening process. And if they fulfill the criteria to be enrolled and there's a spot open, they will go ahead and receive the treatment.
Great, thank you. I think I'd like to go back a little bit to what we were talking about before about needing the international cooperation. And certainly to Dr. Hexey's point about needing to join forces, and we need that sophisticated approach to work internationally, as stated by Professor Anderson. So is there a specific CAR T-cell pediatric network that exists today? Are there therapeutic groups that are working together internationally? And if not, do we need one?
Good question. So my understanding is that there is not an international collaborative group. There are some groups working together, at least in the U.S. Unfortunately, there was the pediatric CINT, so Cancer Immunotherapy Network, funded by the NIH, that was supposed to run multi-institutional cell therapy trials, immunotherapy trials in the U.S. And unfortunately, that organization is no longer moving forward. There are some foundation-based setups, but I think that in my view is that it's going to be hard for a single center to enroll large enough patients fast enough to have conclusive data, as the number of eligible patients is shrinking. Fortunately shrinking, because they are, being cured at a higher rate from upfront therapies. So I think this is something that the field in general should consider and seriously, work toward. One thing is that, every center has their own therapeutic approach. So how to then prioritize within such organization, I think, is always going to be a challenge. But perhaps, setting personal, incentives aside, and that would be important for the field.
So I can just add to that. So there is a very newly launched team that we're a part of. That's through the Cancer Research UK, partnered with the NCI, and it's called the Cancer Grand Challenge. So it's a very newly formed team that was just announced a couple months ago. So that'll be at least across different sites in the US and the UK.
Great.
Dr. Anderson, anything to add there to your points about creating that international approach?
Yeah, I had to drop out and miss some of the discussion. What I think probably I've little to add to what's already been said. It's really important, I think, that individual trials and new drugs effectively are done at very high standards, probably in small centres. That's the sort of cost-effective way to do it. And then international collaboration is required when there is something to evaluate in a larger number of patients. The number of variables is so huge, the complexity of making the products and the virus and the manufacturing is so enormous that it's unrealistic to have a combined agreed international approach. However, what's of paramount importance is the sharing of data at a very early stage, as soon as it's appropriate, and especially within the field, so everybody learns from each other. And then within the large trials organizations like COG, SciPen, ITCC, to have a joined up approach once we get to a stage of needing to evaluate in larger groups of patients.
Certainly sharing from the patient and family perspective, being able to break down those barriers and share and work collaboratively, especially from that international level, is critical and highly important to patients and families. So whether it is how we figure through those priorities to be able to create a push from the patient and family perspective, we would say we'd certainly love to see that happen sooner than later.
Yeah.
In terms of if CAR T-cell therapy is a breakthrough for neuroblastoma, that gives us great survival rates. Do we see that production can be commercialized, can be sped up? Are there ways that you're already seeing this as a possibility?
Yeah.
Shall I say something?
Sure.
Sure, go ahead.
Seeing as I was off for a while. Yes, so I really think it can be. Obviously in the leukemia fields, this transition has occurred with some products which are now been transitioned into pharmaceuticals, albeit at great cost. So it's still important from a more global perspective that an eye should be had on how these could be made more affordable, and not just affordable by the very rich in the future. I think there's maybe two areas of solutions. One is improved technology and higher throughput manufacturing. And the second is maybe a little bit more science fiction, which is off-the-shelf products. So the development of allogeneic therapies, it's not as, there's been a lot of interest in it. The idea that you can make cells invisible to the immune system. I really think the technology has quite some way to go in order for the generation of cellular platforms which can be used between individuals. But that is not to say that it won't eventually become something which is actually viable.
Yeah. I would just want to add a comment that I completely agree that right now we are in this kind of transient stage where we are making products for each patient individually. I think, as John mentioned, off-the-shelf approaches will be coming out. There is some ways to figure out how to evade the immune rejection. And then, of course, not in that distant future, I think in vivo genetic engineering is going to take on. I think, we've had the luxury of engineering cells outside of the body because we can draw blood and do it. But if you look at some other regenerative medicine technologies, where it's liver storage diseases or muscle disorders, people have been already working on engineering cells inside the body. There are tricks because lymphocytes proliferate, so integration of the gene is required. But there are several, actually companies now working on this, and really important publications that I think is going to dramatically reduce costs and hopefully improve accessibility, not too distant future ideal.
I can just add that another type of off-the-shelf product that the field is pursuing is, rather than giving the T cells themselves, is giving a protein product, a bispecific T cell engager. So, where one end of the molecule can recognize the target and the other end of the molecule recruits the patient's own T cells. So it really can potentially cut out the whole cell manufacturing process and be able to use the same receptor still, but at a much more scalable manner.
Absolutely. Certainly recognizing that, this is still a health technology in its very early stages for solid tumors. When you, sort of maybe wave a magic wand or think ahead many years into the future, we've talked about the GPC2, the GD2, the FOX2B, the NKT, the PC, in terms of these different CARs, do you ever foresee them having a potential of working together or attacking from the inside and the outside simultaneously? When you sort of imagine what might happen in the future, are those things that you're already thinking about?
Yeah, I think we should. Yeah.
Absolutely. Yes, we're doing exactly what you just said.
Yeah, agreed. Yeah.
Great. So then the next question that I had here was then, are the doctors on this panel considering collaborating with each other in any upcoming trials and work?
Not in any upcoming trials, but who knows? I hope that we will cross paths sooner or later, and then we can work on the same specific objective very soon in the non-distant future.
Yeah. And as Mark mentioned, the Cancer Research UK Cancer Grant Challenge is designed specifically to generate clinical trials which are at least coordinated internationally, even though for sort of logistical reasons to do with regulations, it's difficult to open transatlantic trials quickly. But within Europe, we are very actively pursuing international collaboration. Yeah.
As you imagine those many years in the future, are there other things that have you excited now about CAR T-cell or gene therapies, and specifically for solid tumors and neuroblastoma?
I think we are overall in a very exciting phase that can go any way. I think aside from just CAR T-cell, advances, there are also a lot of advances of doing liquid biopsies and tracking the progress of patients. Because in leukemia patients, that's much easier since you have most of the time access to the tumor cells since they're circulating in the blood. With our solid tumor patients, one of the things that really hampered the progress was that you can't really justify to biopsy patients unless it's clinically indicated, right? So with these new technologies like cell-free DNA, on a micro scale, like doing deep genomic sequencing and all of that, that will tremendously help our biologic understanding and push the field forward. So overall, I think it's very exciting.
Professor Henderson or Dr. Hexley or Dr. Yarmarkovich, would you like to have any final words with that question?
I
think it's a very Go ahead, Dr. Anderson.
Apologies. Yeah, I think one of the things we have an eye on is other cancer types in children. So the brain tumors for sure, and certainly the sarcomas and things like that. So I think that's one of the areas I'm most excited about.
Dr. Yarmarkovich?
Yeah, I was just going to say that I think this panel represents kind of a snapshot of all the exciting strategies that different groups are pursuing in the CAR T-cell field. And I think a few years down the line, we'll have a lot more information on what the promising approaches are, how to kind of consolidate them and combine different strategies together. And I think, that hopefully will lead to a rapid evolution in the coming years.
And Dr. Hexie?
Yeah, I mean, I think it's a super exciting, time in research, and with the ultimate goal that it should benefit the patient significantly as we move forward. There are several approaches that, now that there is really strong evidence for CAR T-cells to work in leukemias, there are several approaches that are being tested for patients with solid tumors, and I think the faster we get the clinical data, the better. And I certainly hope that once we figure out how to effectively treat patients with relapsed refractory disease, maybe these approaches will be moving to the up. We talked about it, but the relapsed refractory patients, many of them go through the upfront therapy, which is not negligible burden. If you think about just the induction, the stem cell, the now tandem stem cell rescue, the high dose chemo, radiation therapy, and maintenance with antibody therapy, this is quite long and burdensome time for the patients and their families. So if that can be shortened, that would be really good.
We all agree with you on that one, absolutely. So Professor Anderson, Dr. Hexie, Dr. Yuramarkovich, and Dr. Nguyen, thank you very, very much for your time today, for your expertise, and for all that you shared with us. Thank you to everyone for watching and for all of your questions. Remember, if you miss a session, it will be available to watch later on demand right here on the event platform. So up next, after the breakout at 1840 GMT, we will have 2 special interest talks. There will be the anti-GD2 antibody therapy, an important approach in the treatment of neuroblastoma, which you can watch on this stream. And there's the post-treatment, a parent's perspective, which will be running in parallel on stream 2, which you can access via the left-hand menu. So thank you very much again, and enjoy the rest of your day.